I'm delighted to introduce Steve, whom we know from last year. Steve's a postdoctoral scientist at EPFL in the group working on the Blue Brain project, where he creates experimental and computational approaches to explain decision-making. Before that, he worked with Justin Gardner at Stanford, where they did fMRI, amongst other things. He studied comp neuro at University of Bordeaux, where he got his master's in medical and biological sciences. And he focuses, amongst other things, on statistical methods. And he can tell you a little bit more about what he does. Steve, if you're ready, over to you.
Thank you very much, Dan, for the very nice introduction. Here I'll be talking specifically about a model of the cortical microcircuit, and I will explain why it is very important to develop techniques to be able to evaluate the neural recording techniques we have today and their accuracy and the reliability of the information that they're providing to us to study the brain. So I'm very happy to be here for the second time. So very specifically, I will talk about why a generative model of high-density neural recordings built on a biophysically detailed model of the cortical microcircuit is a very useful tool to ensure that we measure accurate and reliable neural information when we look at spike trains. Single unit recordings and classical low-density electrode arrays are now giving place to high-density neural recording techniques, such as the first Neuropixels probes, which was released in 2017. Neuropixels is currently the only technique that enables the simultaneous recording of the extracellular electrical potentials of thousands of neurons at the high spatial and temporal resolution of single neuron and single spike, and at multiple spatial scales within brain areas and across brain areas, allowing the study of contextual modulation of early sensory areas by long-range feedback projections during decision-making, which is my subject of interest. It also enables the study of information encoded at multiple temporal scales, ranging from milliseconds to days. A very good example of this and of the power of Neuropixels is in visual neuroscience. Most electrophysiological studies in visual neuroscience are focused on only a subset of visual areas. Fewer than half of them have been well studied. For example, V4 or MT involved in motion perception, while most areas, for example, V3A or PEO, have only been sparsely investigated. Neuropixels technologies make it possible to perform thorough and systematic large-scale surveys of neural representation across all visual regions, accessing both the superficial, but also the deep brain structures. For example, using Neuropixel, Troutman et al. Very recently have shown how neurons' visual receptive fields shift in an orderly way for stretches of approximately 1 millimeter, as displayed on the graph to the right. They showed that these shifts both reflect the topographic and retinotopic representations in visual cortical areas. Okay, take-home message so far is that Neuropixel technology is very powerful to basically map visual representations in the brain, but extracting spike trains from Neuropixels recording requires spike sorting. And the problem is, and this is a critical problem, spike sorting is biased. Puccino and colleagues have shown that when evaluated on Neuropixels recording simulations, where spike neuron assignments are known, Kilosort, the state-of-the-art, correctly detected most neurons, represented by the red bar, but displayed 3 types of biases. It created false positives, which are spurious neurons not actually present in the simulation, and this was the most common bias. It created redundant neurons by splitting the spike trains of a true neuron into several portions assigned to separate sorted neurons, which was less frequent. It created overmerged neurons by combining the spike trains of several true neurons into a single sorted neuron, which rarely occurred. And these results were consistent across 6 of the most recent spike sorters. So left uncorrected for bias sorting hides neurons' true firing rates. It creates spurious neurons and spike trains, which leads to erroneous claims on neural coding and computations. I invite you to consult Hill et al. In 2011 Journal of Neuroscience to see how it can impact visual receptive fields. So how accurately neurons are sorted have been shown to depend on spike properties. Petitari et al. Have analyzed the 12 most recent spike sorters, and they have shown that sorting accuracy increases when neuron firing rate increases, when neuron spike amplitude increases, when neuron spike spatial extent decreases, and Magellan and colleagues have shown that it increases when spike amplitude increases with respect to the background noise. But if manual correction of spike sorting biases, which is what have been done so far, is feasible with few electrodes, it becomes nearly impossible with high density probes that record massive volumes of data. So more accurate spike sorters need to be actively developed to automate and ensure the reproducibility and objectivity of the process. To do that, until now, spike sorters are evaluated and compared on artificial benchmark datasets. The field has developed 3 main types of ground truth to evaluate sorting accuracy and characterize spike sorting biases. First, entirely synthetic ground truth, where simulated noise with realistic but simulated spike waveforms are superimposed at known times. 2, hybrid ground truth, where synthetic or manually curated spike events are added to the background noise. And paired recording dataset, which are the gold standard, but laboratory acquisition of such recording is difficult and time-consuming, demanding simultaneous paired extracellular and cell-attached recording. Because of the very limited realism of this generative model, we don't know how much the evaluated accuracies and characterized biases hold on real datasets. Because these models do not comprehensively reproduce, first, spike shape variety caused by the cell's diverse morphoelectric properties and their location dependence. I will talk about this in a few slides. The second aspect is that they do not reproduce comprehensively the spiking statistics and dynamics per cell type and cortical layer, and the cortical microcircuits large scale, involving 30,000 cells. Fortunately, Isbister and colleagues have recently released, less than a month ago, the most biophysically detailed model of the rat's primary somatosensory cortex. The model was extensively validated against experimental data. It accounts for cell morphoelectrical properties, cell density, layer organization, synaptic connectivity, and has in vivo scale, next to 4.2 million neurons. This is an example of the model's spontaneous propagation dynamics in the primary somatosensory cortex, and of the dynamics evoked by a localized stimulation of thalamic fibers here in the same map of the primary somatosensory cortex. So we asked, what are spike sorting accuracy and biases on a comprehensive, realistic model of high-density recording? And what is the true firing rate distribution of the cortical microcircuit model? To answer these questions, we simulated high-density recordings in a microcircuit of Isbister's model and checked the realism of our recording traces and spikes against experimental data. We then sorted spikes and neurons with Kilosort 3, the state-of-the-art, and by matching our sorted firing rate distribution with experimental datasets distribution, also sorted with Kilosort 3, we could infer the true firing rate distribution of the cortical microcircuit model. Finally, we evaluated sorting accuracy and characterized biases by comparing sorted and ground truth spike trends. And we recovered the cortical microcircuit model's true firing rate distribution by finding the ground truth firing rate, which sorted firing rates matched experimental sorted firing rates. Okay, so first we simulated recordings at the coordinates of the electrodes of virtual Neuropixels probe inserted in a simulated cortical column of Isbister's model. We reconstructed two Neuropixels probes. First, a 32 electrode linear probe with densely spaced electrodes illustrated here by the white dots, and the original Neuropixels 1 composed of 384 electrodes as described in the introduction. We evaluated sorting on the cells within 15 microns of the electrode, which is delimited here by the red rectangle that surrounds the electrode, consistently with the maximal distance for accurate sorting proposed in Buzsáki paper, a recent paper by Buzsáki. In Isbister's model, we model neurons as multiple compartments, N, in which currents I, N at each time step T flow. We sum all these currents to generate extracellular potentials, which contribute to the extracellular field potentials recorded at the coordinate of an electrode here. The generative equation for the extracellular field potential recorded at each electrode site is given by the equation described in Einvall and colleagues. The electrical field potentials are produced by the sum of all compartments currents divided by the norm term here in the denominator, describing the decrease in the field potential amplitude as a function of compartment distance to the electrode sites here. Sigma is the extracellular conductivity that classically converts current intensity to electric potential. This generative model has been shown to account for how field potentials generated depend on neuronal morphology and positions of the synapses and recording electrodes. For example, when synaptic currents are injected in its distal branch, the red circle here, the spherical shape of a layer 4 stellate cell in the cortex produces a symmetrical distribution of field potential on the left and right side of the cell. And this is due to the interaction between the excitatory return currents elicited by the current injection at the point of the circle here in the most distal compartments to the right of the panel and the cell's spherical morphology. In the space surrounding a pyramidal cell, though, the current injection in the neuron's apical branch, illustrated also by this little circle, will produce a bipolar asymmetric distribution of the receptive field, which is due to the interaction of return currents and the neuron's bipolar morphology. So now to the results. We propose Horvath and colleagues open dataset of extracellular recording traces. They were recorded from anesthetized rat somatosensory cortex, and we display here an example of 1 second trace from one electrode. Our generated traces qualitatively matched Horvath's traces. They display the same 3 components: high amplitude to noise ratio signals corresponding to single neuron spikes, lower amplitude multi-unit spikes, and background noise with similar amplitude relative to the other components. Our traces also match the experimental datasets quantitatively. Our traces power spectra are very close to those of 2 experimental datasets, as you can see on this plot of raw power spectrum densities displayed on logarithmic scales for visibility. In 2 experimental datasets described by the black and the gray power spectrum densities, frequency amplitude are near constant up to 1 Hertz, and then scale down linearly with log frequencies as has been documented. Our model's power spectrum density, which is depicted in red, follows the same distribution, although it starts to scale down at higher frequencies. Simulating 7 cortical column, which is more realistic, in brown, slightly improved the qualitative match to the experimental dataset. Both our models match the 2 datasets better than the raw traces from the most recent synthetic model we could access, illustrated in blue. So this is a model by Buccino and colleagues published in 2020. The traces are typically high-pass filtered to discard low-frequency signals and preserve the signals above 300 Hz, those of spikes. Our models also better match the experimental dataset here too, compared to Buccino's model. the amplitude distribution of the trace affects the quality of spike sorting. Here we show that the distribution plotted on the y-axis of the amplitude of our model's traces relative to the noise of the trace, that is the mean absolute deviation of the traces, was within the range covered by the two experimental datasets displayed by the gray line on the top and the black line on the bottom, with our data being in red and brown. Again, our models provided a better match than Buccino's model. Notice that we are on a log-log scale.
Steve,
using clustering.
In the interest of time, I think we've got maybe 3 or 4 minutes, so there could be a bit of time for questions, please.
Okay, so I will just try to compress or go fast. So I will go quickly through certain slides and we can have questions maybe. So basically, now we looked at the spike shape waveforms, and using clustering techniques, there is a team, Polten and colleagues, that have shown that there are 5 types of spike waveforms. This is conserved in cats also. And we also observe the same waveforms in our data. I will skip that. It explains how spikes shape depends on their location to the electrodes and also morphoelectric properties of the cells. The take-home message here is that here we also observed in the data the same dependence on the spike shape relative to the location of the soma of the cell and the electrodes around that cell. I will skip that. And furthermore, when we sum all the contribution of different cells for the some extracellular recording cells signal, we also see something that had been documented before, that the signal, the spikes, disappear within about 50 to 60 micron from the soma, illustrated in red here, and completely disappears within 200 micron. This has been documented on the left by Buzsáki 2004. I will skip that. I described it. We looked at the firing rate distributions to see whether they matched also real data. And what we see in red and brown dots, these are our simulations, that they are well fitted by log-normal distribution, the lines, but and this is an alignment with documented firing rate distribution in Buzsáki and Mizuseki 2014. And they were within the range of experimental dataset distribution on the left in black and on the right in gray. I will skip that part. So finally, we looked at the sorting accuracy and biases on our model. And to do that, we compared ground truth neuron spike trains with sorted neuron spike trains. What we see is that, what we showed, is that 4 ground truth neurons out of 500 isolatable neurons, that is only 1% accuracy, were accurately sorted or well detected, with more than 80% match with a sorted neuron, compared to 80% typically documented on other ground truth. The majority of these neurons, of the ground truth neurons, shared only 10% of their spike with at least a sorted neuron, which is very bad accuracy. And other neurons were either poorly sorted, overmerged, or oversplit, or they were completely missed. The missed neurons were only sparse neurons in black, while all detected neurons, the only 4 detected neurons, were active neurons. In conclusion, I hope that I have convinced you that our model extracellular recording traces, spike shapes, and firing rates are realistic. On our model, the state-of-the-art spike sorter accurately sorts 1% of isolatable neurons compared to the 80% typically documented, overestimate the true population firing rates, and poorly sort the sparse firing neurons, entirely missing 20% of them. This is just my acknowledgement slide to my collaborators, to my team, and also to all the people that have provided very insightful feedback. Thank you.